Okay, hi guys, welcome to Biology Online with Henny van Vieren. Hi Year 12s, I hope you are really well. Today we'll be going further and looking at the 2019 paper for genetic variation and change, and specifically question 3, where we will talk about the platy, which is a live-bearing fish, which I absolutely love. I have some of them in my aquarium at home. And they mate, and then they have babies. They don't lay eggs, which is awesome to watch. My kids love it, and then you have this little breeding colony, which is quite cool. But you have to then think about getting new migration in by going to the pet shop and buying other platies, so that you always make sure that you have new alleles coming in, otherwise the inbreeding will take over. Anyways, without further ado, let's get into it. Okay, so level 2 Maya 2019, question 3. So these are the little platies I told you about. You can see this is a male because they have quite a different little fin over there compared to the females, because he uses that fin to then guide the sperm into the female's body, which is, yeah, it's really cool. He swims backwards while he's doing that. You can tell I like these fish. Sorry. The southern platy fish, Xiphophros. Wow. Xiphophros maculatus is a freshwater fish native to North and Central America. It is easy to keep in an aquarium, so it has become a popular aquarium fish amongst breeders. A gene that codes for the pigmentation pattern at the base of the tail fin is found at the P locus. Now, initially I read this P locus, I'm like, what? Am I supposed to know what a P locus is? No, that P simply means pigmentation pattern, okay? I don't know why they did that, but so it's just the locus. On one of its chromosomes, there are 8 multiple alleles found at this locus. So in real life, there's 8 different alleles. And interesting, this one is called the Mickey Mouse, because you can see it's got the 2 ears and the big head. However, we're going to be looking at the crescent spot and the dot spot. Anyways, so now the first part of the question, A, was describe the term multiple allele, and because you studied your definitions, you know, and you knew how that you need to write, multiple alleles are alleles of which there are more than 2 alternatives available at one locus, or more than 2 alternatives available for one gene. Okay, and we always use blood type to look at that, but here's another example. The picture below shows some of the tail spots, or the allele patterns, expressed by the platyfish. All alleles are co-dominant except the no spot allele, which is recessive to all the other alleles. So this is the no spot allele, and they just told us that it's recessive, but they still give us this capital P plus, which I find strange. It should have been, in my opinion, a lowercase p, but hey, that's what they want us to use, so that's what we'll use. The crescent spot found on the tail is pattern crescent, and then we have over here pattern dot. Alright, so dominant, and this is recessive. But when these are together, it'll be codominant. So the picture below shows some of the traits, patterns. All the alleles are codominant for except the spot, which is recessive to all other alleles. Okay, so we got an A already. Woo. Now, explain the difference. Remember, explain is when we go into merit territory. So this is a merit question. Explain the difference between codominance and recessive inheritance. Now, because you studied your definitions, you know that codominance, because remember, whenever they ask us a question, we start with the definition. So we describe codominance for achieved. Codominance is a form of dominance where the alleles of a gene pair in a heterozygote are both fully expressed in different areas of the animal. Remember co, and we remember it as the cow codominance is the cow dominance, where we have the black and white cow. You can see both areas dominant, but they are not in the same areas. Otherwise, it would have been incomplete dominance, and that is when you have a white cow mating with a black cow and they have gray babies, because incomplete means both of them are in the same cell fully expressed. Okay, incomplete dominance is both are dominant in the same cell, whereas codominance is where they are both expressed fully but in different areas of the animal. Now, recessive inheritance. So recessive inheritance is when the recessive phenotype is expressed only when there are two copies of the recessive allele. We actually describe that usually as complete dominance, where we said complete dominance is when if a dominant allele is present, it will always be expressed, and you will only see the recessive phenotype. It is only expressed when there are no dominant alleles present, and it is a homozygous recessive allele or homozygous recessive genotype. Anyways, so for that you got another two achieved. So just like that, you have passed this question already. Now, then we go to explain. We're going from, we're still busy with the same question. So remember, we're busy with explain the difference between codominance and recessive inheritance. Now we're moving on from not just mentioning it, so we're going from achieve level, where we just describe it. Now we're explaining it. So codominance is a form of dominance where the alleles of a gene pair in a heterozygote are both fully expressed. This results in a phenotype that is neither dominant nor recessive. In this case, 3 possible phenotypes and 3 possible genotypes. Can you see they're referring back to our example? Okay, and that is very good. When you're referring back to your example, you always want to, that's how we aim for excellence. Okay, so we're explaining codominance. So if you were just describing it, you would just explains means what it is and how it works. So codominance is a form of dominance where the alleles of a gene pair in a heterozygote are both fully expressed. Bang. That is what it is. Do you agree? Read it again. Do you agree? Yes. Now, how does it work? Well, when you have them, this results in an offspring with a phenotype that is neither dominant nor recessive. In all 3 cases, you have 3 possible phenotypes and 3 possible genotypes. So this one is going to be a crescent, so fully crescent, which is that one. This one is going to be crescent and dot, so not sure how that will be expressed. That might be the Mickey Mouse. And then this one is dot dot. Okay, which obviously is the dot. And recessive inheritance is when a recessive phenotype is expressed only when there are 2 copies of the recessive allele. Bang. When you stop there, you stopped at achieved level, but now we need to move on to merit level. The dominant allele is absent. There are 3 possible phenotypes with 3 genotypes. And that one is going to be crescent, and this one is going to be dot, and this one is going to be no dot whatsoever. Okay, or no spot rather. And those are 3 merit options for you there, or 2 merit options actually. So they also then further went on, and now we can do our Punnett Square. So if fish breeder crossed 2 fish that were both heterozygous for crescent and no spot. So they were both crescent and no spot, heterozygous. Remember, hetero means different. Okay, homo is same. This one is hetero, so it has to be different. Complete the Punnett Square below and describe the expected genotype and phenotype ratio. They always like to start by putting the heterozygous. So basically they put the individual here and then they say what type of sperm and egg can they make, and that is what you put in the second little circles. Okay, so obviously can give one allele to this sperm and one allele to that sperm, and this mom can give one allele to that egg and one allele to this egg. Now you know how this works. We basically drag the across and we drag that one down, drag that across, drag that one down. So with this first one we'll have, it's both going to be crescent crescent. Okay, so that is homozygous crescent. So we know they are going to look homozygous crescent. In this case, it's going to be heterozygous. But remember, this is recessive, so it's going to be heterozygous crescent. This one is a heterozygote as well, but it's heterozygous crescent. And this one is homozygous recessive or no spot. So our expected genotype ratio is 1 homozygous dominant, 2 heterozygotes, there they are, and 1 homozygous recessive. Now remember, this is for every fertilization. This is not when you have 4 fish babies that they will follow this pattern. It is per fertilization. And our expected phenotype ratio is, when we just look at the phenotype, crescent, 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 and no. So that's 1, 2, 3, and 1. So you always have to remember that you need to write the words. 3 crescent spots to 1 no spot. Do not just write the 3 to 1, okay? You have to write 3 crescent with 3 crescent spots and 1 no spots. So if you had the Punnett Square correct, you would have gotten an achieved, and if you had one of these 2 correct, you wouldn't got another achieved. So there's already 4 achieved opportunities for you guys. Easy peasy. Okay, so if we look at the second pod, because they wanted us to do 2 Punnett Squares, they said the fish breeder also crossed 2 fish that were heterozygous. Remember, hetero is different, homo is same, so hetero, different, for crescent and dot. Complete the Punnett Square below and describe the expected genotype and phenotype ratios. So, complete the following monohybrid Punnets. Now we put the whole genotype in there, and the whole genotype in there, and then we look at the gametes that they can form. So for the sperm, the dad can only give a crescent there and a dot there. The mom making an egg can only give a crescent there and a dot there. So then we do the actual Punnett Square, and you can see this is homozygous crescent, this is heterozygous, but remember it's codominance, so you're going to see both crescent and dot. This is heterozygous and then homozygous dot. So our genotype ratio is 1 crescent, 2 heterozygotes, and 1 dot. Okay, homozygous dot. But our expected phenotype ratio is going to be 1 crescent with a spot. 1 crescent spot, 1 crescent and dot, and 1 dot only. Okay, so this is our crescent, this is the crescent and dot, codominance, both expressed in the same organism, and this is homozygous dot. Righty, so if you got the Punnett Square correct, you got an A. If you got this or that, you have another A. So there's now 6 achieved options for you already, opportunities. However, if you got both Punnett Squares correct and both genotypes and phenotype ratios correct, then you got a merit. And that's a very easy merit to get, as long as you remember to write out the words and put in the genotype ratios. Okay, so then we move on to the excellence part of this question, and the reason why we know that it's excellence is because it starts with the word discuss, and excellence is always discuss or compare, and it's where we go that little bit further. Okay, so we know that when we answer any biology questions, we always start with the definitions. So discuss the genotype ratio for the above. Um, discuss why the genotype ratios are the same for the above crosses, but the phenotype ratios are different, and the advantages of multiple alleles to a population. So describe. Describe is achieved level. I'm sorry. So firstly, they talked about genotypes, they talked about phenotypes, and they talked about multiple alleles. However, multiple alleles they defined for us in the question, so we need to tell them what a genotype is. A genotype is a combination of two alleles present for the same gene in two homologous chromosomes. Very straightforward. Phenotype is the physical appearance of the offspring or trait coded for by the genotype. Very easy. Okay, there's two achieved levels again. Now we're going to move on to explain. And remember, when you see the word explain, this is merit level. So explain why the genotype are the same but the phenotype are different. And remember, they wanted us to discuss this. So the genotype ratio for the crosses are both 1 to 2 to 1, because every one of those two Punnett squares, we're crossing individuals that are heterozygous. And if they're all heterozygous, your genotype ratios are going to be the same. However, the phenotype ratios are different because the alleles are expressed differently. Okay. So explain means what it is and how it works. Okay, what it is and how it works. But as I told you, when you get in biology and you're going for the excellence level, then you say, so what? So what the phenotype ratios are different because the alleles are expressed differently. How are they expressed differently? Well, and then you start discussing codominance and how that works, and recessive dominance. Recessive inheritance, sorry. Now, explain the advantage of multiple alleles to a species. When you have multiple alleles, it means you have lots of genetic variation. There you go. That's an advantage. So what? Well, remember, lots of genetic variation gives us species survival advantage, but that is going towards the E part of it, and we'll look at that in the next slide. And then give one example of the different phenotypes the different allele combinations give for the heterozygous phenotype. Give one example of the different phenotypes. Remember, we're looking at the Punnett squares. Give one example of the different phenotypes the different allele combinations give for the heterozygous phenotype. So, if you then decided to talk about the codominance one, remember we said codominance. Co is like the cow. Cow dominance. Like our black and white milk cows. Cow dominance. They are both black and white at the same time, but in different areas of the animal. That is codominance, cow dominance. Okay, please remember it that way. Whereas incomplete dominance was that we have the incomplete, it is in the same cell, and if they're in the same cell and they are both dominant, so if you have a black cow and a white bull and they have a calf together, if both the black and the white are expressed in the same cell, that is incomplete dominance, and then you will have a grey cow. But this is not the case. Over here we have codominance, cow dominance, so we will see both of the heterozygote phenotypes in the offspring. So that's what we said here. In codominance, you see both the homozygous genotypes expressed in the heterozygous phenotype. So, in other words, both a crescent and a dot. Okay, and then explain all the possible. So then you also talked about when we had the dot and the recessive. Okay, and because that is recessive inheritance or complete dominance, the heterozygote will be no spot and dot. Okay then. So that was 3 opportunities there. Well, 4 opportunities for merit. Easy peasy. Okay, and now discuss why the genotype ratios. So it's still the same question, but now we're moving on to the discuss part of it. So we've already said the genotype. So again, they want to know why were the genotype ratios for both of them 1:1, but the phenotype ratios were 3:1 and 1:1. Okay, so you've already said this for merit. The genotype ratio for the crosses are both 1:1 because each Punnett square is crossing individuals that are heterozygous. However, the phenotype ratio are different because the alleles are expressed differently. So what? Well, one Punnett square shows recessive inheritance or complete dominance. That means that you will have 3 crescent and 1 no spot. And that is because the heterozygous, I would have loved to see that in here, the heterozygous are also displaying the dominant trait, whereas only the homozygous recessive will display the recessive trait. The other Punnett square gave codominance 1 to 2 to 1, which meant that you have a homozygous dominant will give you a crescent spot. The 2 of them that were heterozygous both have crescent and dot, and the one that was homozygous dominant dot is a dot. Okay, so and then we already, so there's one excellent opportunity. And then if you discussed the advantages of multiple alleles, so we already said that the advantage is genetic variation, and we said, so what? Well, the more different phenotypes you have, the more those phenotypes can respond to natural selection pressures. In other words, things like environmental changes and so forth, and increases the chance of survival of a species, especially if environmental conditions change. Can you see that literally those 2 lines there make the difference between a merit and an excellence answer? Okay, then we move on to the third part. Remember they said here, and talk about the advantage of multiple alleles, but also they compare recessive and codominance. So over here we haven't compared them yet, we just discussed them, and now we're going to go and compare them. In codominance, cow dominance, remember, heterozygous genotypes is different to both of the homozygous genotypes. In codominance you see both the homozygous genotypes. In other words, you see both dominant alleles expressed in the heterozygote. For instance, the crescent and the dot. We've already said that. So it depends on what alleles are present as to what phenotype is expressed in the codominant phenotype. Codominant heterozygous phenotype. So crescent and no dot will be crescent and no spot. Crescent and dot will be crescent and dot, and then dot and no spot will simply be dot. So we took this back, we already said that, but now we're taking it back to the example and making sure that they see that we're applying our knowledge over here. We go on, we say, so the phenotype appears as a result of the combination of the alleles and not the presence of a dominant allele, as with recessive inheritance. So in other words, they are both dominant at the same time, but being expressed in different areas of the animal. That is how I would have answered this question. This is compared to recessive inheritance, or in my opinion, we could have said complete dominance, where you do not see both possible phenotypes expressed in the heterozygous, because the one is dominant and the other one recessive. Yes? So only the dominant one, homozygous dominant genotype. I don't know why they do that. Only the dominant genotype, the dominant allele will be expressed, and the other one is completely recessive. Hence you have 3 genotypes, but only 2 phenotypes. And the dominant phenotype result from both homozygous and heterozygous phenotypes. Okay, you see how we took that a little bit further and we just unpacked it more. Brilliant, another E for you. So easy peasy, guys. I really hope that it goes well, and I will chat to you again soon. Keep well. Thank you so much. Bye.